Next speaker is Michael Kiebish, who has been interested in cardiolipin for many years and has made several contributions to it, and who has also worked on the mouse model, and he's going to give us his side of the story. Michael.
Thank you. So first of all, I'd like to thank the Barth Syndrome Foundation for giving me the opportunity to speak today, as well as for funding to do part of this research. So today I'll be talking about my work when I was a postdoc at WashU School of Medicine, really targeting cardiolipin as a molecular therapy to treat Barth syndrome. And during that process, I actually discovered some unknown findings of potentially what the mitochondrial lipidome might actually be doing in health and disease. So now I've actually migrated back onto a pharmaceutical company called Berg Pharma and Berg Diagnostics, so obviously I have to put a disclaimer that all this work was done at WashU School of Medicine. So I'll be briefly going over some of the cardiac, lipidomic, metabolic, and adaptive mechanisms embodied by the Barth syndrome mouse model. I won't be going into a lot of detail because we have several talks, and I'm sure it'll be covered again and again if I continue to go over that. But I'll also be talking about, specifically in regards to specific aspects, molecular targets that actually can attenuate what we see defective in Barth syndrome. In particular, was a mouse model I used that actually transgenically expressed cardiolipin synthase in the heart. So by merging these two mouse models together, we could actually test a molecular mechanism by attenuating bioenergetic dysfunction and lipidomic dysfunction. And also I had the opportunity to cross the mouse with IPL2 gamma transgenic mice that express in the heart, as well as IPL2 gamma knockout mice, to actually determine the role of IPL2 gamma in cardiolipin remodeling in respect to tafazin. And I looked at obviously the lipidomics and subsequent survival effects. Because obviously all these combinations will have either a different biological effect or a different effect on survival of the mouse. But overall, the goal was to really interpret the mitochondrial lipidome and bioenergetic connection that actually could be defined as a therapeutic target. And in my research, I tend to do multi-omic integration studies, integrating global shotgun lipidomic analysis, gene microarray analysis, bioenergetic endpoints, and actually a new thing, signaling lipidomic analysis, or mediator lipidomics, what's actually the biologically potent molecule and physiological function. So I like to actually connect changes in the mitochondrial or cardiac lipidome with what might be liberated by a phospholipase to then be enzymatically oxidized to a very biologically potent molecule that might control vasodilation, vasoconstriction, and calcium homeostasis. And by integrating more of a global understanding in different models of different lipidomic disorders and different mitochondrial disorders, you can actually get at the heart of the pathophysiology. So this is just a shotgun spectrum of the cardiac phenotype in a mouse that was knocked down from conception and now is 2 months of age. And obviously see in shotgun approach, you see a lot of different peaks. It's not as clean spectrum like Fred Vaz showed for just cardiolipin. But cardiolipin is over here, and when you knock down the gene, you see a dramatic decrease in the 0.5 isotopolog of cardiolipin. But we also see an increase in the knockdown of monolysocardiolipin. And one thing I'll continually emphasize, we see an increase in dilysocardiolipin. And it seems to only be in the mouse since it's never really been found in the Barth syndrome phenotype. So I would really encourage any people who do have the mice and they're able to actually measure it, I would love to see if you actually find it as well. Because we have found this close to 100 times. It always seems to be consistent. And if you actually quantify the molecular species, you obviously see a dramatic decrease in tetra-18-2 cardiolipin in the knockdown mouse, which is in the white bar, and the wild type are in the black bar, as well as an increase in monolysocardiolipin and specific species of dilysocardiolipin. And obviously we do global lipidomic analysis. We see a lot of other changes that we'll talk about later in PC, PE, and lyso-PC. But if you do mitochondrial function assays, such as state 3 respiration, you see differences in substrate utilization. And I presented some of this work at the last Barth syndrome conference, but I'll kind of relate it to why we are using the other models to target this approach. We see decreases in palm-carn stimulated state 3 respiration, no change in pyruvate stimulated respiration, and an increase in glutamate stimulated respiration. And that was obviously of significant intriguement to me, since usually things don't go up. And after chasing this, I wanted to know why glutamate was stimulated. So to link the drive of glutamate stimulation, I measured adenine nucleotide translocase activities, which bring ADP into the mitochondria and bring ATP out. And for some reason, whether it's the lipidomic or other adaptive mechanisms, ANT is substrate specifically driven in the Barth syndrome mouse model, about six-fold greater on amino acids. There definitely seems to be a selectivity for amino acid utilization for mitochondrial function. And if you knock down the mouse model in different ways where you knock down a mature, you actually get some differences in what substrate are actually utilized. So this mouse model definitely has differences in bioenergetics depending on how you knock it down. Now, in terms of analyzing electron transport chain activities in the knockdown, the mouse that was actually knocked down in utero, we see complex 3 enzyme activity deficiencies and slight complex 5 enzyme activity deficiencies. But we don't see a decrease in ATP production in these mice. So I'm not sure what these actual enzyme activities are actually doing in terms of causing a mitochondrial defect. There obviously is some compensatory mechanisms. Now, one thing I want to really emphasize that really drew my interest and I was actually working on for about a year and a half is integrating oxidized lipidomics. Now, for some of you that aren't as familiar with it, different fatty acids that are polyunsaturated can be oxidized enzymatically at specific locations. And these molecules have very potent differential biological functions. And most commonly known is arachidonic acid, which can be oxidized to form heats, and prostanoids. These affect vasodilation, vasoconstriction, and inflammation. But one of the things that isn't as well characterized is oxidized linoleic acids, which can be formed into dihomes, hodes, oxodes, and epoxomes. And these can control calcium influx and immune response. And also the oxidized DHA molecules, such as resolvins D1 and D2, which are very potent anti-inflammatory molecules. So upon looking at the lipidome of Barth syndrome, I found dramatic changes in PC and PE, which contain arachidonic acid, and 22,6 acid, as well as 18,2 and the rest in 2 positions. So it seems like there was definitely a suggestive nature to actually look at these oxidized molecules. But before that, I had actually been working with IPL2 gamma knockout and other models that control mitochondrial lipid turnover and found that mitochondria actually produce a lot of these oxidized molecules. I looked at human heart. So mitochondria actually is a source of the production of these oxidized molecules, which can actually control some degree of cardiac function. If you take mitochondria out and you give them just a little tiny bit of nanomolar and micromolar calcium, they produce hundreds of folds more of these oxidized molecules. So the mitochondrial lipidome actually is a scaffold to generate these and is mediated through different enzymatic pathways and different phospholipases. And upon analyzing the Barth syndrome cardiac oxidized lipidome, and black bars are the wild type and TAS are the knockdown, we actually see a selective decrease in the oxidized molecules for 9-hode, 9-oxoode, and 9,10-epoxome. So a specific pathway for selectively oxidizing linoleic acid, whereas the other molecules aren't really selectively oxidized. So obviously we see a liberation or a lack of a liberation of certain molecules which will be turned into these very biologically potent molecules. In terms of oxidized DHA, we only see the most well-studied oxidized DHA, the resolvins, which are lower, which are the most potently anti-inflammatory molecules, whereas the other DHA molecules were not lower. And overall, DHA content is lower in these mice. So we see a selective oxidation. In terms of heats, eats, and prostanoids, we see a differential change in heat production. Some are up, some are down. So we see differential regulation. And we also see no increase in the cardioprotective eats that are well studied. And in terms of prostaglandins, we see overall differential expression, especially LTB4 and thromboxane 2. So there definitely seems to be a disorganization of these oxidized molecules in the Barth syndrome phenotype. These are, well, they're free fatty acid, and then subsequently oxidized through enzymatic means. Upon analyzing the microarray analysis of these mice that were induced conception at 2 months of age, we found differential regulation in specific pathways of genes, specifically for amino acid metabolism in specific amino acid metabolism and synthesis, intraconversion, GTP hydrolysis, purine metabolism. But overall, it was a very protein-centric changes that actually painted a different picture than just lipidomic abnormalities. We also saw a lot of solute carriers, things that would bring metabolites in and metabolites out, as well as ion homeostasis. About 27 different ones differentially increased. So definitely the lipidome is changing the capacity to bring different metabolites and ions. Downregulated was actually the breakdown products of amino acids and different pathways and valine leucine and isoleucine degradation, which actually mirrors a lot of the metabolomic analysis of amino acids. But overall, this model actually just demonstrated the lipidomic abnormalities discovered in the boys and young men with Barth syndrome, except for the dilysylcardiolipin, which I'd like to see someone else confirm. Also displays altered substrate utilization, decreased fatty acid utilization, and increased glutamate-stimulated respiration. It demonstrates altered or disorganized mediator lipidomic signature, which can affect physiological function. And it demonstrates compensatory enzyme kinetics, such as adenine nucleotide translocase and changes in electron transport chain activity, as well as changes in gene expression. With that being said, at the same time, they had been working on a very unique mouse model called the cardiolipin synthase transgenic mouse model, which was transgenically expressing cardiolipin synthase in the heart of the mouse. And this was actually a very unique tool because it allowed me to provide a molecular therapy to treat different abnormalities in cardiolipin that would be precipitating mitochondrial dysfunction. And our assumption that we would be making more cardiolipin. And obviously we characterized the mouse in a recent JBC paper, and we found that we had 6-fold increased CLS activity in mitochondria isolated from this mouse model. But we didn't have an increase in total cardiolipin content. And it probably has to do with the biomembrane kinetics. You probably can only pack so much cardiolipin into mitochondria, you really distort the cristae. In terms of tetra-18-2, we actually found a 40% increase in tetra-18-2 cardiolipin. So we found selective turnover and hyperremodeling in this mouse model, which actually I determined to be a very useful tool to understanding the role of tetra-18-2 in bioenergetics and potentially signaling. Upon analysis of other lipids, and I'll only talk about PC, we found differential changes in the molecular species which had 18-2 in the SN2 position, 22-6 is lower in the SN2 position, and arachidonic higher. So we're seeing the scaffold for signaling lipids change. And again, it might be more of a location thing. Where these things are liberated from phospholipases might have to be in specific molecular species to be orchestrate this other physiological process. So by turning over 18-2 and turning over cardiolipin, we might be changing 18-2's cycle through more longer chain polyunsaturated fatty acids, which might displace their role in the scaffold for signaling. And what I found very interesting was that the cardiolipin synthase mouse actually makes more of this 9-hode, 9-oxohode, and 9 and 10 epoxome, which was deficient in the Barth syndrome mouse, as well as 1 or 2 others increased. With a much lower 22-6 content in the mouse heart, we only see a selective few oxidized DHA molecules that were lower. In terms of arachidonic acid metabolites, we see a dramatic increase in heats, a significant increase in heats, the cardioprotective heats, and a increase in specific prostanoids. So definitely, and not the TXB2, which is altered in Barth syndrome, or the LTB4. So we see definitely a differential expression of these oxidized metabolites just by controlling cardiolipin turnover and reorganizing the lipidome. Now in the CLS mouse model, we actually see an increase in complex 3 activity, whereas in the Barth syndrome, we saw a decrease. So we see a different compensatory mechanism. But also we see a decrease in complex 4 activity. But this is not leading to lower ATP production either. So this reorganization of electron transport chain was not really a defective mitochondrial effect. And in terms of measuring the ANT activity that brings ADP in and ATP out, we saw a substrate-specific, slightly lower measurement of glutamate-stimulated ANT activity. As I mentioned before, Barth syndrome had a dramatically higher ANT activity. So by controlling enzymes that control the lipidome, you can control other enzymatic effects that can regulate metabolism. So overall mitochondrial respiration in this mouse model had a lower glutamate-stimulated respiration, which might be able to compensate for what the increased glutamate-stimulated respiration in Barth syndrome, had a lower palmcarn, which wasn't defective in terms of ATP production, normal pyruvate, and normal pyruvate or lower pyruvate and glutamate. In terms of microarray analysis, there wasn't really much change in terms of any pathways. Maybe one amino acid metabolism pathway in tRNA and one downregulation of a hemoglobin pathway. So nothing pathogenic really coming up. So obviously I crossed the Barth syndrome mouse model with a CLS transgenic mouse model and induced the transgene to see what if it affect. If it would increase tetra-18-2, would it rescue, or would it tell me something completely different? And the goal is to obviously look at the lipidome, look at the bioenergetics, and look at the survival of this particular mouse model. And that would allow us to tell if cardiolipin synthase could be a very novel target for treating Barth syndrome. And I'll just kind of anchor you here. Blue is wild type, TAS is orange, white is CLS, and TAS crossed with CLS knocked down for 2 months, which was efficient to knock down and bring in monolysocardiolipin. Actually had a lower tetra-18-2 cardiolipin. So that was we had a 50-50 bet of which way it was actually going to go in the lab, and there was definitely money riding on the table. And this kind of also anchors us in determining where tafazin actually plays a role in cardiolipin biosynthesis because of the actual molecular species you actually determine. So TAS is here, CLS is here, and actually we have lower tetra-18-2. In terms of the other molecular species, and there are a lot more than what I'm presenting over here. In lipidomics, you have to only show what you can. You can't show all 60 molecular species. 18.1, we have the double cross mimics the Barth syndrome mouse model, and more longer chain carbon, the double cross also mimics the Barth syndrome model. So it seems to be across the board. But intriguingly, when analyzing monolysocardiolipin and dilysocardiolipin, we see tri-18-2 monolyso reflecting of the decrease in tetra-18-2. But we actually see some monolysocardiolipins that are more representative of immature cardiolipin, containing 16.0, 18.1, 18.1, 18.1. So these do exist, but by putting selective biological pressure on the metabolic pathways, you can actually see that the biological process of lipidomic remodeling does occur through these pathways. Because you have to imagine lipidomics is a temporal change, and you're only really getting a snapshot, so you have to really tease the system to see it. Now, in regards to location, since PC has been really primarily the driving force of the remodeling substrate for tafazin, we wanted to look at particular molecular species. And this can really show interest to what is actually being harvested for remodeling. 16.0, 18.2 is the same in the double cross in tafazin, but 18.0, 18.2 is dramatically increased in the double cross, which had a lower tetra-18-2 cardiolipin. So we definitely see a differential here in what species are being picked. 18.2, 18.2 is unchanged. So either it's being turned over at the same rate or not being targeted for adding 18.2 into cardiolipin. Thus, these molecular species might have a differential affinity for actually remodeling the tetra-18-2 or 18.2 enrichment in cardiolipin. 22.6 containing species are dramatically lower. They kind of mimic the CLS. So I think actually these species serve as a scaffold to put 22.6 on there, and that's why they're deficient. If they're not being removed for modeling, you're not going to be able to place these species over there to balance it out. So we're definitely seeing an interconnectivity in the lipidome between organization and molecular species. 22.6 in this species, 18.2, 22.6, is dramatically lower in both Barth and the double cross TAS CLS. And arachidonate-containing species are increased in both CLS and the Barth model, which might be indicative of changes in different signaling lipids. Now, in terms of bioenergetics, which is a key question I was really interested in, blue again is wild type, TAS, CLS, and yellow is a double cross. The double cross is slightly lower than the Barth syndrome, and not statistically different, but it's completely rescued. But it definitely did have a lowering effect on glutamate. It definitely didn't go up, even with more or less tetra-18-2, but I wouldn't say it's rescued. In terms of pyruvate metabolism, this is more of a mature knockdown model. The Barth syndrome mouse model in the mature knockdown has lower pyruvate. CLS is normal, and the double cross TAS-CLS is in between. So we definitely have a differential in bioenergetic regulation. If you measure complex 4 activity using ascorbate and TMPD, you see that the double cross mimics the Barth syndrome model and not the CLS model. So we see a differential in bioenergetics. So although we see less remodeling in this double cross, we don't see a worse bioenergetic phenotype. And unfortunately, I collected all these tissues for also the signaling lipidomic analysis, but it just haven't been run yet because I would like to see a balancing of what signaling lipids are being generated and what are deficient in the Barth syndrome. So those hopefully will be done in the coming weeks. And I think that also the other important thing is, as I mentioned, if you take the doxycycline off the mouse model, you see the lipidomic and bioenergetic return after about 2 months back to normal. And if you actually have CLS overexpressing in its mouse model, and I did a lot of dynamic experiments collecting mice every 15 days to look at the lipidome and bioenergetics, you see a 4-fold increase back to normal by overexpressing CLS. So you actually see some therapeutic value of targeting CLS, getting the lipidome and bioenergetics back to normal. Now, one experiment I did before I actually left WashU, I actually did a lot of experiments, but I actually wanted to challenge Richard Gross to say, how is IPL-2 gamma really involved in cardiolipin remodeling? So I did a double cross experiment, which obviously takes some time, where I crossed both. I wanted to see both ways. If you transgenically overexpress IPL-2 gamma, and if you knock it out, what does cardiolipin look like? And I know a lot of members in the audience have actually looked at phospholipases as a therapeutic value, and more recently, I think Dr. Schlamme published a paper looking at IPL-2 as a therapeutic value, looking at cardiolipin. And I wanted to see what the lipidomic effect was. And I was actually quite surprised to uncover what the effect was, and find out what the survival of these mice actually were. So the goal is obviously to see if it attenuated cardiolipin dysfunction and its survival. So what we have here is blue is the wild-type mice again, orange is TAS crossed with gamma knockout mice, and TAS is in purple, and TAS crossed with gamma transgenic mice in the heart is in green. And what we find is that the TAS mice look exactly the same as both the IPL-2 gamma transgenic and the IPL-2 gamma knockout in all the molecular species. But what was different was when you do these double cross, when you analyze monolysocardiolipin, you see it only affected the 18-2 rich monolysocardiolipins, tri-18-2, 18-2, 18-2, 18-1, and 18-2, 18-1, 18-1. The other monolysocardiolipins it did not affect. So there is definitely some selectivity, both overexpressing and knocking out, because you're obviously affecting both the phospholipase activity on the PC, but also preventing the removal of acyl chain from cardiolipin. But even more surprising to me was the dilysocardiolipin had the most change. So it was a dramatic lowering of dilysocardiolipin. And if you actually look at the return, just to get on a different point, the dynamic, if you take the doxycycline off the mice, you actually see dilysocardiolipin disappear first, then monolyso. So it definitely seems to be a very important biosynthetic pathway which phospholipases such as IPL-2 gamma have a role very importantly in dilysocardiolipin, which we might not be able to see, and then monolysocardiolipin to remodel. But the species you actually generate are not 4-chain or normal species to normal level. So to conclude, I want to emphasize if you knock down the Barth syndrome model in utero, you definitely see a lot of lethality, probably 60%. I mean, we already kind of went over it in one presentation. I think it was Colin's presentation. You definitely see lethality. If you knock it down in more of adult mice, maybe 5 out of 40 you might lose. When you actually have the CLS cross with tafazim, I only lost 1. So there definitely seems to be a slight protective effect. If you knock it down, the gamma knockout cross with tafazim, 3/4 of the mice die. So you actually need gamma to keep this mice alive. It might actually be part of the oxidized signaling molecules. The gamma transgenic cross with TAS mice, they all live. They do great. It didn't have any lethality. So definitely there's an underlying, even though cardiolipin might look the same, there's an underlying other mechanisms that actually been working at play to control the physiology. Thus, by using these transgenic models and dynamic flux and different time courses, you can actually understand the lipidomic and bioenergetic connection and understand potentially pathological lipidomic species as well as therapeutic lipidomic species that you can actually use as a therapy. And the regulation of the mitochondrial lipidome may actually hold unknown mechanisms to go well beyond structure and function. And like I said, with the oxidized signaling lipids, if you give some of these oxidized signaling lipids to mitochondria, they sing. They can handle calcium, millimolar amounts of calcium. The MPT doesn't open. And they also choose different substrates to burn. There's really an unknown, unexplored territory of how the lipidome can actually kind of self-regulate some of the mitochondrial functions. And it's there primed and ready to go. But the most important thing I really want to emphasize is this inducible tafazin SHR knockdown is really a very powerful, invaluable tool to actually investigate these molecular mechanisms. Without it, we wouldn't be able to understand the biology, understand the pathophysiology, or even understand mammalian therapeutic mechanisms to target Barth syndrome. With that being said, I'd like to thank the Barth Syndrome Foundation for funding and support, as well as all of you. NIH as well for my mentors lab in Washington School of Medicine. Dr. Todd Cade, who was really a good resource to have someone else who's working on Barth syndrome at Washington School of Medicine. Dr. Jinlin Han, Richard Gross, Kuei Yang, and Jinping Liu at Washington School of Medicine. And all of you for your attention.
Thank you, Michael. Papers open.
That was beautiful work, Michael. Really, I mean, we could probably talk for the rest of the conference, you and me, just about our data. And just a question I wanted to confirm. Early on when you showed production of the oxidized lipids, was that from mitochondria?
This was actually the heart, but we've also confirmed it in mitochondria too.
Okay, and so have you confirmed just blotted for the presence of cyclooxygenase, lipoxygenase enzymes in mitochondria?
No. That's why I think this ER mitochondria connection is very important in a lot of things, and it seems to be underappreciated. And we call this kind of mitokine channeling. So we think they're released from the mitochondria channeled to either the ER or other mechanisms, and those are actually like the important biological connections which control these processes. And we've given the drugs to inhibit these, and they actually restore the same thing as the product itself.
A lot of work and beautiful work. So when catalepin synthesis was cloned from mammal, from human, the speculation was that catalepin synthesis does not have ACO preference, But your work showed that there is an increase of tetrolinolenic acid, after you generate transgenic mice. What is the heart performance? It's, you didn't touch upon that. Is the heart is function normal? Oh, they have dilated cardiomyopathy.
They function normal. We've done SDZ studies with these mice. They don't have echo abnormalities. Their bioenergetics are maintained. Their lipidomics are maintained. They actually have a little better ejection fraction and Tay index too. But we haven't really gone into too much of the physiology because you don't usually make things better than nature makes them. So it's hard to interpret when things get better than wild type. It's actually very difficult to interpret.
Michael, nice data, real good work. This phenomenon you described, that glutamate ANT activity was much more elevated in case of the Barth syndrome cells. How do you explain that? And first of all, how did you do that actually? How do you measure the ANT activity? Two questions.
So the explanation, I'm not sure if it's how the lipids are actually integrating with the specific ANT activity, or there might be, I'm sure if we did proteomics and saw different isoforms or different post-translational modifications, there might be something going on there. For doing the ANT activity, you get the mitochondria to basically have state 3 respiration, and you titrate carboxyl attracticide at small dosage to see it go down, and you measure the curve of the functional ANT.
Okay, yeah.
I'm interested in the glutamate observation in treating the boys with Barth syndrome. Obviously, the goal is to increase alpha-ketoglutarate, and glutamate, or what we use in clinical medicine, is glutamine to increase alpha-Kg. And I haven't used it because glutamine tends to run high to begin with. But if it's the same in humans, that sluice is open, then maybe glutamine, which is very well tolerated compared to some of the other amino acids, maybe it'd be worth trying that again, or trying it in Barth in a way I haven't so far.
I definitely wonder if it's a compensatory mechanism or it could be a therapeutic mechanism, and it's hard to tell if you're going to push it over the edge one way or the other, depending.
Okay. Would you mind explain more about double knockout with PLA2 gamma? Because 3 kinds of monolysyl quadrilepin decreased. Why?
I think it has to do with the selectivity or PLA2 gamma's control over dilyso, making it go. So if it is a metabolic pathway between dilyso and monolyso, or if there is acyl selectivity with the phospholipase, it might regulate which species are actually generated or which species are turned over or hydrolyzed.